I'm Jonah Comstock, Editor-in-Chief of pharmaphorum. We're here on day 2 of ASCO 2026, and I'm joined by Teresa and PK from the Takeda team. I'll let you two give your full names and titles so I don't mess them up.
Sure. No, absolutely. Teresa Bitetti. I'm the president of the global oncology business for Takeda.
And I'm PK Morrow. I lead oncology development at Takeda.
So give me the headlines, the overview of this year at ASCO for Takeda. Is it a lot of papers? Is it really impactful papers? What are you guys excited about talking about this year?
Well, you know what? Let me start by saying that our strategic goal for Takeda is to build depth in 3 tumor types, which is thoracic, GI, and heme. We have a very long history in heme. We've been a leader there for a long time, but we've now really bolstered our pipeline, not only with more heme assets, but also with solid tumor. We did a recent deal with Innovent, and so we have 3 assets coming through that deal. And so we're excited because we have data. I'll turn it over to you, PK, maybe to share some of the data that we have that we'll be sharing here at ASCO.
Absolutely. So thank you so much. So let me start with the partnership with Innovent and the fact that we're presenting and have 3 posters here at ASCO. I'll start with the frontline data, which is being presented at ASCO, and that is related to our dose optimization data in which we randomize patients in the frontline setting with non-small cell lung cancer without actionable genomic alterations to one of 3 doses of a drug or a program called TAK-928. What is TAK-928, you will ask? TAK-928 is a first-in-class bispecific that targets 2 areas. One is the alpha receptor of IL-2, activating the tumor-specific T cells, and secondly, also targeting and blocking PD-1. By that combined mechanism, we have potentially an IO-IO bispecific that acts upon the tumor cells in a very orthogonal way. And what we've seen in the frontline setting is that is therefore translated in the clinic to very encouraging findings with the 3 milligram per kilogram followed by 1.5 milligram per kilogram dose, demonstrating encouraging efficacy in the frontline setting, very tolerable safety, as well as the ability to continue to dose in the long term. In addition to that, we also are presenting data in the second line setting in patients with non-small cell lung cancer with the same program, TAK-928. In this case, we are also showing encouraging findings which are consistent with a potential long tail, as we like to see in those curves, with a long tail of progression-free survival as well as overall survival. Our third poster is primarily a trial in progress for TAK-928. I'd be remiss if I didn't also talk about the fact that we have some publications also across our other marketed products, including Frizocla and many others, as well as Rusfertide, which Teresa alluded to. We're developing and anticipate a potential PDUFA in August of this year. And that in that case, we are reviewing or demonstrating data about the poor adherence currently to treatment with PV, demonstrating a clinical unmet need for better and therapies in PV, as well as the fact that with the treatment of Rusfertide on top of other therapies, we're able to actually reduce the utilization. 42% of patients are able to reduce or discontinue cytoreductive therapy in the presence of Rusfertide, reducing the risk of polypharmacy.
Yeah, so it's actually, I'll just add right on that, we're very excited on the lung data because not only can this molecule be used as mono, but in combo, but it's also addressing the IO-resistant population, second line. So IO resistance, I don't need to tell you if you've been in this business a long time, there's a huge unmet need, and we haven't seen the kind of efficacy there that we would like to. And now with TAK-928, it's very promising, the data that we're seeing. And we also know we've had over 1,200 patients treated with TAK-928. So we're very excited about the potential of what we can do there. And of course, we have the upcoming launch of Rusfertide. So.
But TAK-928 it's still investigational, right?
Yes, that is correct.
But it's so many patients. So it feels like you must be close to the finish line, or how is that going?
Wait, you want to speak to the 1,200, but it's been in large, different cohorts on various tumor types that it has been tested in.
Absolutely. So along the lines of how you've seen IO therapies being investigated, across solid tumors, what we really want to do is better understand the efficacy and safety profile of 928 across. So we're disclosing the non-small cell lung cancer, but beyond that, there are multiple solid tumors in which we've been investigating in partnership with Innovent the efficacy, and that's why you're hearing that number being so significant.
Yeah. Well, I am hearing a lot of companies are sort of adjusting their strategy to try to um, get into, initially come to market in multiple indications at once because of
Yes.
the IRA. Is that part of what's happening, or is this just sort of like the pathway that makes sense for this drug?
It's truly honestly a data driven decision. So it is actually the fact that we want to bring, we all know that cancer doesn't wait, so we want to bring this therapy to patients as rapidly as possible. The critical unmet need, to be very frank, is actually in the second line setting or a later line area, but also we want to continue to advance and bring the best therapies potentially to patients in the front line setting.
Yeah, yeah, and we're also looking at this in microsatellite stable CRC, which again another area of high unmet need that has been traditionally more of a cold tumor. And what we've seen, the synergistic immune activation for this PD-1, alpha biased IL-2, it's promising. It's exciting.
So to take us back to where we started here, Teresa,
Yeah.
you were saying that Takeda's larger strategy
Yes.
here is to move from heme into more solid tumor types.
Yes.
Talk to me about how TAC928 and all the other data at ASCO this year fits into that larger
Yeah.
strategy.
So, thank you for that question. Right. So for the three pillars, thoracic, GI, and heme, we've got a good pipeline. Rusfertide about to launch. We've got a Rotacept in the pipeline. We have an option on imatinib, and so we're excited there. What TAC928 does for us is in the thoracic and in the GI. So in thoracic, we already have a Lumbric on the marketplace, but this will allow us to enter into non-small cell, both squame and non-squame. And then when you look at the GI, we already have Fruzaqla on the market for CRC. This enables us to enter into CRC. And in addition, we didn't go in as much detail on our ADC for Claudin 18.2, which has a topo1 payload, but that will allow us to address pancreatic as well as gastric cancer. So our expansion into the solid tumor has really developed with these addition of these assets. And I don't need to tell you how fast the oncology world moves. And so what's always wonderful about ASCO is absorbing all the data that's coming out, and then it allows us to ensure that our development plans are moving as quickly to match where the standard of care is going or where the combinability might be with other assets. And so, as we look forward and look at our development plans, this is where a conference like ASCO is kind of fantastic, right? Because you just get a bird's eye view of everything that's happening in space.
I'm going to ask each of you to tell me a little bit about the trends you've been seeing and hearing, at the conference so far, and what you're looking forward to hearing. One or the other or both.
Okay.
You start on that.
Personally, I think we all agree that ASCO is a premier conference in which we're really seeing the merging of science, innovation, and patient-centric drug development all merge into one. And so as I look forward to ASCO, a couple things. One is we're really, to be frank, very excited about the ability to share data, on TAC928, because what we're seeing is that the trends in development are new and innovative ways to address the IO landscape. So, building upon the previous PD-1, which are truly monoclonal antibodies, as you know, PD-1 monoclonal antibodies, and now moving into potential PD-1 or VEGF, as well as PD-1 IL-2 alpha, as is ours bispecifics. What we're seeing is orthogonal approaches in order to help to overcome IO resistance, as well as to move and provide even greater efficacy in the front line. So very excited about that. The other element I think that we're seeing is a lot of ADC development currently, right? With potentially dual payloads, potentially newer targets, and greater efficacy with a debulking strategy. So those elements are very exciting. And of course, we always talk about the fact that we were interested in areas where previously there areas that were previously undruggable, I might say, that now are being addressed in more fulsome form to address critical unmet need, like, for example, in pancreatic cancer.
Yeah, no, I think you just said it all.
All right. Well, in that case, thank you both. Thank you both so much for chatting with me today, and I hope you have a great rest of your show.
Wonderful. Thank you
Thank
for the
you.
time. Thank you.